Okay, guys, let's get started. Okay, hi everyone. My name is Shomo. This is week 4 of NGS Data Analysis and Applications, course offered by Professor Iman Dhar of IIT Kharagpur, and I'm the PMRFTA from IIT Bangalore. So, today primarily we'll discuss about alignment, different algorithms for alignment, different data structures used for alignment, and uh, how we tackle certain issues that we come up uh, for alignment during genome alignment. So, yeah, we'll focus morely on the different types of algorithms and how these different types of data structures are made for so much big data sets. For example, the human genome. So it's a very big data for one particular genome of a human cell. It's going to be a very big data set. So how do we handle that? How do we use certain techniques to compress our data? Because without compressing, the file will be so large, so it cannot be properly used to do anything with it. And also, since the amount of data is the sheer volume of the data is so big, we also have to look at the space complexities of the different algorithms as well. How much space does a particular algorithm take, and also along with it, how much time does it take? So we need to find the balance between space and time so that we can have an ideal algorithm that will not take too much space, also um, complete the alignment procedure in a much shorter period of time. Okay, so let's get started. I'm starting at 5 p.m., but nobody has joined in the meeting yet. So, like, if so, welcome everyone who is watching later. So, yeah, let's get started. Okay, so first we need to define what the alignment problem is actually. So, like, what do we mean by alignment of DNA sequences? We'll primarily talk about DNA sequences, but similar metrics can be applied to protein sequences as well. The issue with protein sequences is like we have around 20 amino acids that form different type of combinations to form different type of proteins and everything. But for DNA, we only have four bases: A, T, G, and C. So it's we'll tackle first we'll talk mostly about DNA sequence alignment, but protein sequence alignment is something that is also done very frequently, and it is quite useful as well. So first of all, before coming what is the alignment problem, let us talk a little bit about why do we need alignment. So what's necessary what's the necessity to do sequence alignment for a particular sequence. So, say for example, we talked about different types of sequences in the first class. So, say for example, you have a sample that has been sequenced by Illumina, and it has given you a set of reads in a FASTQ file. Now, what you know about the reads is basically uh, j- just have the reads actually, nothing else. So you don't know where the reads came from in your genome. You just have a reads and its sequence. So you have a 150 base pair read with its sequence, but you do not know where in the genome did it come from. So primarily the alignment is done to tell you where the read is originating from in your in your data, but we always need a reference for an alignment. So we need a pre-constructed reference that is already been there for for us. There are different versions of these reference genomes. So currently the newest one is the T2T. Previously to that there was the GRCS38 reference for humans. So we have these different types of reference genomes, and we use these reference genomes uh, for for sequencing. And how do we use that? We use this reference genome as a reference to tell us from where did our reads originate, and what is the difference in our reads compared to the reference genome at that particular position of the genome. So it can tell us about mutations, it can tell us about different type of variations in the genome, and everything. So it is a very important step in handling of NGS data. So the first part of the lecture we talked about different type of sequences. Then we talked about how we do after we generate the reads, how we do different type of pre-processing like uh, removing adapters, checking quality of the bases and everything. And after that, the step that comes after that is sequence alignment. After we have cleaned out all our data, all our reads, now we want to find where did this read originate from, and does this read vary in some way or form compared to the reference genome, so that we can make certain inferences from it. So that is why alignment is necessary. Now, this is why alignment is necessary. Now, we need to define our alignment problem in a proper way. So, what what is our alignment problem? So we have a read, we have a reference, a big reference, and we want to find where that read came from the reference. So uh, imagine like there are two protein or DNA sequences given by these two strings X and Y, capital X and capital Y. So capital X, the sequence of capital X is like small x1, small x2, dot 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 till small xn. Same with capital Y, small y1, small y2, dot 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 till small yn. So basically, uh, this basic. So since in DNA we only have four ATGC, only these four base pairs we have. So uh, it's a finite number of alphabets that can be used to form the sequence. So for DNA sequences, the alphabet is ACGT. For protein sequences, there are 20 alphabets like this. Uh, there are more actually, like depending on the amino acids. So protein is made up of different amino acids, and these amino acids form these peptide bonds within them, and they form a long chain of amino acid, which is the protein itself. So your sequences look something like this. What you want, so this will be your input, two DNA or two protein sequences like this, strings of sequences. This will be your input. Now what do you want as your output is basically the optimal alignment between these two sequences in the form of a list of columns of types. So basically, is y the jth element of y, so the so your y is like y1 y2 till yn. So the jth element of y, does it match with the ith element of x or not? So in form of these type of columns, so either it matches, xi matches with yj, or xi does not match with any of the elements of the sequence in y, or y does not match with any of the sequences in x. It can be either or, like it can be uh, like either. So it can match. It can be a deletion in the second sequence, or it can be a deletion in the first sequence. So if you consider these two sequences given by capital X and capital I, capital Y. Now, if you're trying to align them, so alignment in a very intuitive manner is just to match these different strings together. So if you just want to manually match these strings together, so as you see, uh, like there's one T matching here in the second position, just like in the second position. Then you have these two GCG here, GC here, so these two match, but this comes at the cost of two gaps here. We'll talk about gaps and how we can handle gaps and everything. But just if you want to do it intuitively, just to give you an example. So GCA match here, but with two gaps. Again, T does not match with C here. Again, there is an C here which does not match here. Then again, CA matches. So if you're given two sequences like this and asked to match them as much as possible, this will be kind of your optimal alignment. But obviously, in biological, when you're working with biological data, there are certain things to keep in mind. So it's just not two separate strings that you are trying to match. These are DNA sequences, and they follow certain rules. So the mutation that you're considering here, this has to follow certain rules. This mutation that you're considering here, that has to follow certain rules. You need to have further evidence than just like matching up the next element. That's why you skip the previous one. So it's not as simple as that, but yeah, this is how we begin. Okay. Now, as I was saying, your reference and the reads that you have generated from your sequencer or your experiment, they will not match most of the times, like exact match, because sequences have diverged. So what happened is, so we created a reference genome, uh, and like that single reference genome, there can be multiple variations in because all humans are different. No two humans will have the exact same amount of same DNA sequences in both of them. There will be some differences. Some particular mutations will be present in one person. Some particular mutations will be present in the other person. So that this will make the two persons as separate, distinct people who have different, distinct features. So these variations are what gives us this kind of features, and like these variations are important in terms of evolutionary biology. So we all came, we all kind of, all our ancestors, if we see our ancestry, we all diverge from a common ancestor. So our common ancestor, whatever sequence that person had, was mutated through multiple generations to reach us and what we are today. So if I compare my DNA sequence with the sequence of my, say, great grandfathers, there will be variations. There will be differences in the two genomes. There will be different type of mutations that has been acquired through the different generations. There can be other mutations that has been acquired through other kind of things, that some sort of disease or anything, and it has been passed on to the next generation as well. So it is not possible to have exact same match for two different humans. So now how do we then, if that is the case, then the question comes, how do we do the matching of one sequence to the reference sequence, one read to the reference sequence? Before that, like, let us talk about different types of variations that you can find inside in the genome. So the first is substitution, where basically a single nucleotide substitution, where where a single base pair A, T, or G, or C is replaced by another, the other one. Say A is replaced by T, G, or C. Similar, T may be replaced by A, G, or C, and like it's just a base substitution. One base was there; it has been replaced by another one. Deletion, it's just a deletion of a single base. It can happen. Insertion, some sequences might be inserted. In this case, we are considering single nucleotide insertion, but you can have big insertions and big deletions as well, like more than 50 base pairs, more than 100 base pairs. These are possible. But for our case, we'll only consider the single nucleotide ones. There can be inversion. The sequences might be inverted. So ATGC might be CGTA. So fully inverted. Right. And transposition and duplications as well. So transposition is basically when when another part of a chromosome, say DNA from chromosome 10, is cut and pasted on DNA, and pasted in the chromosome 1. So this is called transposition on, and duplication is basically duplication of a particular sequence multiple times. So it like increases the it basically amplifies a particular sequence inside the genome. So these are the different type of variations that you can have. These are not always like, these are some of those variations. You can have much more complicated variations as well. But to go with, these are the most commonly found variations that we see in the genome. Now, like, the question comes, what happens if the single nucleotide deletion or insertion occurs in the coding portion of the genome? And what happens if a single nucleotide substitution occurs in the coding portion of the genome? So what will happen is basically, since these are coding portions of the genome, from this part the mRNA will be formed, from that the mRNA protein will be formed. So if there are substitutions or deletions in the coding regions of the genome anywhere, so it will hamper the downstream elements as well. So that means the mRNA and the protein. So this can happen, and this is This is very common in genetic disorders and different kind of diseases and everything. There is always some sort of this variation that has been occurring, which is actually not good for the for the person to prevent the disease or something like that. So these are the basic three for our cases. These are the basic three types of variations that we'll find. That it will be a match or a mismatch. So it can be a match, direct match or a mismatch. It can be a deletion. So there is no match or mismatch. It's just a blank here, and there can be an insertion as well. So if something is inserted in X, it will not match to Y. So it will not match. So if there is an insertion in that particular portion of the genome in your sample, what you will see is the reads will align, matlab, a single read. This part of the read will align to the reference, this part of the read will align to the reference, but in the middle part, it will not align anywhere near the alignment of these two parts. So what it means is this part has been inserted from somewhere else. So this part does not belong in this portion of the genome. So majorly in these three types of sequence variations will come across. Okay, now as we saw, okay, like it's the alignment problem is not that straightforward. There can be variations in the genome, and so you need to have a certain type of scoring system to tell you which is the best alignment, because considering every type of mutations, insertions, deletions, substitutions, whatever be the case, there can be multiple alignments of of two particular sequences. So like it's not that the two sequences will align only in one way. It can align in like multiple different ways if you take into consideration different type of variations within the genome. So you what you need is a scoring scheme which will tell you, okay, out of all those possible alignments, which one is the best one. So this is an example of that. So alignment scoring schemes reflect biological or statistical observations about the known sequences and are frequently represented by scoring matrices. So this is what is known as a scoring matrix. So this is your this is one of your sequence, this is another of your sequence, and you have different scores for different matches. So if I'll come to the scoring in just a minute, but yeah, so you'll have a scoring matrix like this when when in one in your basically column names you'll have a sequence, and in the row names you'll have another sequence, and inside the matrix you'll have the scores between the pair of the sequences. Now here how it is defined for this particular case, you can divide it in your own way, whatever suits your purpose, but here how it is defined is basically if A and A matches like this, it's plus one. If xi and yj are matched, so if two particular elements of a sequence match, it's plus one. If there is a mismatch, you consider that as a penalty and you deduct minus one. And if there is a gap, so in these cases like insertions or deletions, if there is a gap in alignment, you consider that minus 2. So this is how it is assumed for this coding system. So let's go through the matrix now. So A and A matches, so it's plus 1. C and C matches, it's plus 1. T and T matches plus 1. G and G matches plus 1. Now A and C doesn't match, so minus 1. A and T doesn't match, minus 1. A and G doesn't match, minus 1. So similarly, rest of the boxes in your scoring matrix is filled again. So if a gap matches a gap, what will it be? So it doesn't matter because there is no alignment of that sort. So you cannot consider gaps in both your reference and your leads. You have to consider gap in either of them. So both of the gaps doesn't make much sense, right? So yeah, this is how the scoring system is done, and this is how the scoring matrix looks like. Now, in general, the gap penalty or the G uh, or minus G, generally the gap penalty is given with a negative. Uh, it is denoted as the G. It and this is how it is calculated. So basically, if you have, say, two sequences like this, there are three A's and a C, and then you have AGC. Now you can align it in many different ways. You can just directly align A here, G, you can consider this as a mismatch, A and G mismatch, and then a C and A mismatch. But if you do it this way, this is another way of doing it, like another way of thinking of this alignment is basically you have a gap in the middle. There is one matching in the beginning, one matching in the end. There's one mismatch in the in the middle, and there's one another gap in the middle. So how will you calculate the score? Total score, you just add up all these matches, mismatches, penalties, and everything. You just add up to get a score. Now, like you'll have different types of these alignments, and you add up the scores to get a value. So whatever is the highest value, that is your whichever alignment will give you the highest score is your optimal alignment. So in this case, there are these two scenarios. So one, you have inserted a gap in the middle here, and on the other there's a gap in the end. And let's see how the scoring works. So AA, there's a match, so this will be a +1. Now there's a gap, which is the gap penalty. You have to put a gap penalty here, so it's -2. So it becomes +1, -2, and AG will be a -1 because it's a mismatch. So +1, -2, -1, and then there is another match in C and C, so this will be +1. So if you add the all the four things up, you get a -1. Next, let's try this type of alignment. So there's you've put in the gap at the end. There's an initial match in the beginning, and that's all. Just check if anyone is joined or not. Okay, nobody is joined yet. Okay, I'm just check the timing because yeah, it's Tuesday 5 to 7. So okay. Anyway, let's continue. Okay. Yeah. So in this case, the gap is in the end. So there's only one match. There are two mismatches here, A and G, A and C. These are the two mismatches, and there is a gap in the end. So if you try to score this alignment, so there's a match. This will be +1. A and G, there's a mismatch. This is -1. A and C, this is another mismatch. This is -1, and there's a gap in the end, which is -2. So -2, -1, -1, -4, and +1, -3. So out of these two types of alignments, so see, it's the same reference AAAC, and it's the same AGC. You are trying to align it in different types of way, these two sequences. So what you ideally how you do it is try all the different type of combinations that are possible, score all the different types of combinations, and then take the best of all of them. So in this case, if you compare these two types of alignments, this is a better alignment than this, according to the scoring system that you have. Now, the scoring system is also very important in terms of how you penalize your gaps, how you penalize your mismatches and everything, because there are some biological aspects to it. So, for example, a single nucleotide polymorphism, or a SNP basically, so a single base change in the DNA, will be much more prevalent than a 50 base pair or 100 base pair change in the DNA. So you need to uh, like set your scoring system in such a way you do not these gaps that you are penalizing, these gaps should have like gap opening penalty, like because it's a, a one base pair change. Yeah, a one base pair change should not be uh, equal to a two base pair or 50 base pair change. Like, the impact should not be the same. So your scoring system should be made in such a way that you consider all these different variations that can be possible, and then you make your scoring system. Okay, this was just an example of a simple scoring system, how you can use that to do your alignment. Now, using this kind of scoring system, you can make brute force algorithms. So what are brute force algorithms? It's basically you just iterate over the whole reference, and you try to find all the possible types of alignments, and then you do the score for each alignment and select the alignment with the maximum scores. Now, so this is a brute force way of doing it. Like, firstly, you're not doing any kind of filtering or anything. You're just checking all possible things that can happen. And the other thing is, so this might work when you are comparing sequences of, say, 15 base pairs, 20 base pairs, even 50 base pairs. But when you are comparing sequences of 3 billion base pairs and with a read of 150 base pairs, it is not very it is not very uh, a good thing to do, because it will take up a lot of time, and also you need a lot of space as well, because you need to store these alignments, you need to then put scoring stuff on this alignment and calculate the scores and everything. So you need good data structure, not good, but very big data structures and a lot of space to do this. So, and if you calculate the time taken, it will take exponential time. So practically, this is not possible to do for a human genome level data set, because it will be very big, and it will be very lengthy, and practically you cannot finish your alignment using brute force algorithms of so big data sets. Okay, so the next thing that comes up is what is known as BLAST. So BLAST is a software uh, that is hosted in NCBI, and it's a very commonly used tool to do sequence alignment, even multiple sequence alignment. And there are different types of BLAST. I'll just quickly show you the BLAST website here. Yeah, the full form is Basic Local Alignment Tool. There are different types of BLAST. You can see, like, you can do specialized searches and everything, and there is protein blast as well, where you can check for different types of proteins and everything. So here you need to enter your sequence in a FASTA format, or give the accession number of the sequence. So these are the different types of blast. This is the nucleotide blast, protein blast, and so on. So uh, just check the name: standard protein blast. This is translated blast. Different types of blast here. So we are mostly looking at the blast 10, which is the standard nucleotide blast. So here you paste your FASTA sequence. Say you take it out from anywhere and you paste your FASTA sequence, and then choose your databases, choose your organism, and from here you can add your organisms, and yeah, that's all. And then you click on blast and it will show you the result of the alignment, right? So this is a online tool that is quite commonly used for sequence alignment, mostly local alignment, and you can do multiple sequence alignment using blast as well. Now let's see how blast works. So generally speaking, BLAST works by comparing a query sequence to a database of sequences. So it has its own database of reference genomes of different types of organisms, and if you specify the organism, it will bring up that reference genome and it will compare your sequence to that particular reference genome of that particular organism. So it uses a heuristic approach to search for similarities in the database, making it faster and more efficient. BLAST performs sequence alignment through the following steps. So here's step number one. The first step is to create a lookup table or a list of words from the query sequence. So we'll look at what a lookup table is. How can you make this? It is doing is some sort of indexing, right? So this is a lookup table that it creates, and uh, this type is called as CD. So it basically chops the whole sequence into small pieces with overlapping elements in it. So like this, you can see this is a protein sequence given by this. query. And what BLAST does is basically it breaks down this into small protein sequences, small amino acid sequences, which can then be used to make the lookup table. And this tape is what is called as CD. So for protein sequences, each generally the each length of the seed is basically 3 amino acid long. And for DNA sequences, each of these seeds is usually 11 nucleotide long. So these are known as k-mers. So the K stands for the number of elements in your sequence. So since these have 11 nucleotides, it's an 11 mer. These Since these are three amino acids, it's 3 mer. So yeah. So this is the first step, breaking down the query into small seeds, which it uses to create the lookup table. In the second step, it searches a database of known sequences to find any sequence that contains the same word as the query sequence. Excuse me. This is done to identify database sequences containing the matching words. So this is your database sequence, and uh, basically you have a database of known sequences and sequences that contain the same words as query sequence. So that's where you go and use these schemas and try to find where these particular schemas are present in your whole database sequences. So as you see here, like if this is your uh, basically schemas or seeds, so in this case this only matches one of the three. Here it matches two of the three, and it's in a preliminary search, so there are two matches within 40 residues. So this is possible, like this is the database that it needs to search. So okay, so in step 3 then it scores the similarity between the matching of the words. So as we discussed how we do scoring, BLAST also uses scoring to score the tell the similarity between the matching of the words, and the matching of the words is scored by a given substitution matrix. So this matrix is basically the scoring matrix what you create. Okay, so if a word, in this case the word is the seed, so please keep that in mind. It's Do not confuse. So if a seed is above a certain threshold, it is considered a match. And the two commonly used substitution matrices for protein sequences are PAM. So PAM is a very widely used matrix for protein uh, sequence alignment and scoring, and BLOSUP, which is the block substitution matrix. For nucleotide scoring, the scoring matrix is based on match mismatch. Like the fourth step involves a pairwise alignment by extending the words in both directions while counting the alignment score using the same substitution matrix. If the score drops below a certain threshold due to differences in the sequence or mismatches, the alignment stops. The resulting aligned segment pair without gaps is called the high scored scoring segment pair, which is your optimal alignment. And along with that, also BLAST calculates a statistical significant value for each alignment. This is called the expected value or the e-value. The e-value represents the probability of obtaining a sequence that matched by random choice. A lower e-value indicates the sequence match is likely to be a result of random occurrence. Hence, lower the e-value, higher is the level of significance. It's kind of very similar to p-value, except here it is called expected value of matching, which is which basically the essence is basically of that of p-value. So it tells you the probability of obtaining the sequence match by random chance. So p-value also tells you something like that. Okay, so this is how BLAST works. So in BLAST you have this query, you break it down into small pieces, then you make a lookup table and search your database, and then you use some sort of scoring metrics, which there are certain matrices that are standardized for scoring metrics, which are one is PAM, one is Blossom. There are other matrices as well, and then it does the alignment and gets the alignment score and tells you this is the best alignment with this expected value, right? Okay. Now this is like basically what happens in BLAST is already your index has been uh, sorry, already your difference has been analyzed, and it has been broken down into small pieces, and you know where these small pieces are in your database. And then so once your query has come, you break down your query into those small pieces and you check where these small pieces are matching from your database. But when you are doing alignment locally, uh, so what a good idea to do alignment for using alignment algorithms is indexing. So what is indexing? It's not a very novel idea. It's very similar to the kind of indexing that we have in textbooks as well. So most books have an index where the word is mapped back to a page where it is found. So it's in the back where you have a set of indices where basically each word, it tells you at what page in the book that it is present in. So uh, this particular type of index is called inverted index. And the word inverted implies that there is also non-inverted or a forward index. The image below explains the distinction between the two. So, so here the word is mapping to where which document it is present in. So for example, the word the is present in document 1, 3, 4, 5, and 7. Cow is present in document 2, 3, and 4, and so on. But the inverted index is something like this. So if you have the document, and, and it tells you what are the words present in that document. The cow says moo, like this. So it, the two different types of indexing, one is the forward indexing and one is the inverted indexing, right? Okay. For what we are interested in, so for in our case is what is its use in alignment. So what we are interested in, searching for the best location of a read in the reference, we will use the inverted index, which we simply refer it to as the index. So to find a pattern in a string using an index, we first need to create the index. To create the index for a string that is a genome, so to create the index for the genome, we need to first extract substrings of length, a certain length, and record where these substrings occur. So since DNA is a very repetitive structure, so you have many repeats of ATGC and many small of the substrings that you repeated across the genome for across 3 billion base pairs. So if you know that this substring occurs in these many positions, so in a way you have indexed your genome knowing that this substring, I know it is present here, 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 and here. So when the substring comes up as a query, you know where to align it to. So there comes the importance of indexing. So it helps you to extract substrings and record where the substrings occur. That is very important. And organize the list of substrings and their coordinates and an easily accessible data structure, which is basically a map. So it's something like this. Say, if you consider the words as the substrings of the genome, it tells you that this substring is present in these positions, or this substring is present in these positions, right? Like this, right? Okay. So that is called a map. Now let's consider this example. So this is your string, which is given by this sequence, and these are its substrings. So like 5 letter substrings which across the whole sequence, or like whatever sequence you want to do. So these are the 4 different substrings. So now that you have created so yeah, so now you have created the substrings, you would want to know where the substrings are present inside the sequence. So for example, CGTGC, this is the substring that is present at position 0, which is offset 0. CCTAC, which is present in position 4. So this is position. So The counting, one thing to note, the the indexing, the index number or the indexing is done with respect to the reference. So this is your reference. So at the fourth position, this one, so we start from 0, 0, 1, 2, 3, 4. So at the fourth position, you have the second substring, which is CCTAC, right? Position 4. Now, position 8 is this position CTTAC, which is present in both position 4 and position 8. Okay? So you can have these kind of indexing, and you can sort them by the keys, whichever, like alphabetically, you can sort them by the keys, and you can store them in some sort of a data structure. We'll come to how we store and how we use that, but you can have, you can like break this the whole sequence down into small substrings, index those substrings, and store them in some sort of data structure. That may be a hash table, that may be something else, but we will look into it. Now that you have created this index, now you have to search it. Suppose now you want to find whether a particular pattern, say this pattern CTAC TTAC, which is basically you can represent it here. It is represented as P. So T is the text, P is the pattern. Okay. So now you want to find whether the pattern is present in the text. To do this, we need to extract the substrings from P. So this is your pattern. You need to extract the substrings from the pattern and check the indices they are present in. And if they are present, extend to see if the entire string is present, is matching or not. Now, this is your basically text. This is your pattern. You break down the pattern into small substrings as well. So you have these small four five letter substrings. You've created these four substrings, and this is your index. Now notice this last substring, which is CTTAC. This is in your text. It is present in position 8 and 12. Right. Now there is one hit at index, one hit at 8, and one hit at 12. Okay. So if you look at, so if you are given a pattern to match it to a string already present, already given. So the string that you are matching to is the text, which you have already broken down into small substrings and indexed those. And now you have given a pattern which you are breaking into those same number of sequence substrings, and you are checking where particular is the substring present in your original text index. So in this case, the last substring is present in your position 8 and 12. Right. Okay. In the previous figure, you saw that trying various substrings from P yields 3 failures and a success with 2 hits. So these are all your substrings. Only one of these substrings is present in your text, right? The rest 3 are not present. The rest 3 were uh, failures, and there was 1 hit. Actually 2, because it's the same substring is present in 2 different indices, but there are 3 failures and 1 hit. Yeah, the trying various substrings from P yields 3 failures and a success with 2 hits. Of course, in our case, the T is very short, uh, the text is very short, and you can easily see that an index for a realistic fragment of DNA can be very large. So, like if you're talking about human DNA indexing, so you have 3 billion base pairs, and you have to break down the whole. So the T length here will be 3 billion. Now you need to break down this whole 3 billion sequence into small substrings, and then you need to index these substrings and store them. So both indexing and storing will require space, a lot of space, and also a lot of time as well. So realistically speaking, it can be very large. So for example, for human chromosome 1, which has around 249 million nucleotides, it will occupy slightly over 12 GB of space if you do indexing like this. In these practical cases, a practical solution may be if you skip certain substrings while making the index, because one thing to remember is your DNA is replicative. So what will happen is like it is possible like one substring is present many times in the DNA. So if it is present too many times, you don't need to keep those as separate substrings and index them. That will be one substring present in these indexes. But even after that, it can be very large. So what you can do is skip certain substrings. So it can be like in a pattern. If you follow a pattern like this, say for example, including only the fourth substring in a human chromosome index, human chromosome one. If you do that, like if you do not take all the substrings, uh, you take like the fourth substring and index them, and you do not index the rest of the substrings. So what it'll do is it will reduce the space required by a lot, and it'll come down to from 12 GB to 8 GB almost. And if you as you can see, finding patterns in the text using indexes require finding values for parameters such as substring length and the interval if you use skipping, like as I said, storing every fourth substring. These values have significant implications to the speed of research, memory use, and most importantly is to the specificity. Because we'll come to specificity what it is, but it is very important to have specificity, to have good specificity while indexing, because if not, then your read can match to multiple places and you don't know which place will is giving you the best, which place the read actually belongs to. Okay, so this is a table that shows different values for substring length and interval affect speed, memory footprint, and specificity of finding. And this is the pattern that is given, uh, and uh, you have to find this pattern within chromosome 1. So if you take a substring of length 4 and you do like skip every three substrings and save the fourth one, this is your indexing time and this is your specificity. You see specificity is very low because the substring length is very small. Now if you increase your substring length from 4 to 7, your specificity goes up, your memory usage also comes down, the search usage as well comes down, but your indexing time increases because you need to extract those uh, those substrings and check. So extracting those substrings will be a little bit more difficult. So if you use 10 substring of length 10, you get a greater indexing time and but better specificity and lower memory usage. Like if you take a substring of length 18, it's more specificity, but if you take a substring of length 30 with an interval of 30, the indexing time is much lower, the search time is also much lower, and the specificity has increased a lot. So finding this balance between the length of the substring and the interval that you are storing it to have a good specificity, that is very important, right? So not only do you want to, so there are few factors in play here. So first factor is what is the interval that you are storing at, that will be like help you save space. So if your interval is longer, you'll have to store store less indices, so your the space used will be less. If you use lesser intervals, the space used will be more, the specificity will also be less. So you have to find that balance between the length interval and the specificity so that you can have a good indexing. Now, what exactly is specificity? Specificity can be thought of as a proxy for the number of times an index hits. An index hit can be extended to a true match between the text and the pattern. So this figure tells you the idea how to what it means basically. So consider this is your text. So it is given like this: time for such a word, and it is connected by underscores, right? And this is a pattern ord. You are trying to find this pattern in the whole text. Now using indexes for this part is is fruitless because the, the it doesn't correspond to a match of p. Here you are looking for a pattern of like ord within this. this text, and the first index hit does not produce a match, while the second one does. Decreasing the number of fruitless hits increases the specificity and speeds of the search. So what will happen is, if you have bigger pattern or a bigger keyword which you are indexing, that will help you to weed out this fruitless hits, because these are like substrings, right? This is not the whole pattern, the whole sequence that you're trying to align. So these are substrings. So you would want the exact match. In the whole sequence, you might not have exact matches and everything, but for the substring at least, you would want as many exact matches as possible. So decreasing this number of fruitless hits like this, so it cannot, although it is matching, O and R are matching, this cannot be a match. There's a gap here, and there's a D here, and there's a proper match elsewhere. So this first hit is basically a useless hit. It doesn't give you anything. It is only increasing your false positives. And the second one is a good hit. So if you can decrease this number of fruitless hits, it can increase your specificity for the pattern matching with the text, because this first hit wouldn't produce a match. It will just take up your time, and it will decrease your specificity. Okay. Okay. So in summary, what we have seen is that we can create an index, which is basically a map which contains the coordinates of substrings from a text T. So you have a whole text, you break them down into small small substrings, and you create an index map. So this substring is present in these positions. The next substring is excuse me. The next substring is present in these positions, and you create index map. Now you can use this map to find occurrences of the pattern P within the text T. The map can be represented in varieties of ways using sorted lists, hash tables, and trees. So this map, this like map of like what is the substring and what is the position of this substring, that can be stored in different types of data structures. So this is a basically a sorted list where you have a list of the substrings and the corresponding indices. You can have a hash table where you can store this pattern substrings and their indices. Similarly, using a tree as well. So these are different types of data structures. This is a list, this is a hash table, this is a tree. So this is not a tree, this is a trie. So we'll talk about what is the difference between a tree and a trie a little bit later. But this is what is called a trie, right? This is not exactly same as a tree, but they're very similar. Okay. So what's important here is that sorted list and especially hash tables, they'll provide you a quick way to find the initial hit, but they're limited by the choice of the substring size. So in this previous specificity table before, that the choice of, you've seen that the choice of the substring size will have profound effects on the performance of the search. So the substring size, as I was saying, is very important, because ideally the best substring size would be the entire length of the tree. You can directly match it, but that is not possible, right? So you need to find that perfect size of the substring that will increase your specificity and decrease the search time, right? And also, also you need to have all this in a smaller data structure format. So in a data structure that will occupy a smaller amount of space. Excuse me. Okay. Okay. Now let's consider the string. Nobody is trying. Okay. Just give me a minute. I'll be back. I was just checking if I've joined the right meeting or not, but yeah, I have joined. But anyway, okay. Nobody is here. It's still represented. Okay. Okay. Now, from all that we have seen, different type of scoring matrices, different type of data structures that you can store, the substrings. You can divide this the whole thing into substrings and calculate their indexes and store them. Now let us look through an example of how actually we do it. Let's consider this text, which is given by this. Now, using the logic from what we discussed today, we can build an index using substrings of length 2 with interval 2, meaning you are skipping every second substring. And that would look like this after sorting. So, say for example, AC is present at position 4, AG is present at position 8, I hope I'm right. AG is position 8, and so on. So these are your all your substrings with of length 2 and interval 2. So there are actually 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 10 substrings you have taken, but in total there are 20 substrings, but you have not taken all the 20, you have taken 10 and created their indices where they are present, what is their offset. Now, if you represent this whole thing, like represent the substring and their position as a trie, right? This is what is called as a trie. So, if you represent this thing as a trie, uh, this would like the mapping would look something like this. So, this is your root. So, this is the beginning of your sequence. So, from the root, you're moving out. So first substring is AC. Right? A, you move to this node. C, you move to this node. So AC, and then this is the node that is representing the AC substring from the root. Basically, you're traversing from the root to the leaf. So these are all the leaves, and this is the root, and these are all the branches. So you're traversing from the root to the leaf, and the sequence, substring sequence is given by the branches. So AC is the substring sequence, and at the end, you store the uh, position of that substring, which is the index of that substring inside that node only, right? So like this, you can do for AG, which is the 8, then AT, 14, then CC, like this. So everything you can, like the representation that you have here, you can represent it like this, which is called a trie. So you have a root and the branches we spell out the sequence of the substring sequence, and the leaves will tell you the position of the sequences, right? Position of the substrings in your original sequence. So this is a form of indexing that you can use, right? This is called a trie. Now note that the trie is the smallest tree, right? So this is not a tree, this is a trie. This is the smallest version of a tree, and obviously trie versus tree is a little confusing, I can understand, but what I'll come to the difference. So this trie is the smallest tree, representing a collection of substrings that has the three properties. Each edge, so this is your graph. Each edge is labeled with a character from a given alphabet, in this case, which is A, C, G, and T. Each node has an outgoing edge corresponding to an alphabet character. So this is a node which is called which has an outgoing edge towards another node, which is the branch, which gives you a corresponding character. So basically, if you start from the root, you move to the next node using an edge, which is the branch. Uh, so from the root to the next node, you move here, so you get an A. You arrive at this node, and you move to the next node, you get at C. If you don't move at this to this node, you want to move to this node. So it's A, you move here, then G, you move here, like this. Okay. So there are three major properties. So edge is labeled with a character from the given alphabet. Each node has a single outgoing edge corresponding to an alphabet character, and each key, substrings of length 2 in the above case, is spelled along the path starting from the root. So this is your root, and each of the substrings will be spelled from one from the way you move from the root of the trie to the leaf of the trie, or any other node of the trie. So it is not the opposite way. So you do not move from the leaves to the root. Move from the root to the leaves, right? So you start at the root. You have a here. Then you move here. It's a C. You have a C here. Then you have a C here. So this is representing CC. This is representing CT, and this is representing GT, like this, right? So these are the three major characteristics of a trie that stores the substrings and the indices, right? Now, this above example shows that we can quickly find positions for a given substring, but it all still relies on a fixed substring length. So the substring length is fixed, but if you have a trie with a fixed substring length, you can easily find out the positions the particular substrings are present in using the indices. Now, you will not have fixed substring length across when you're trying to index uh, using indexes. You might want to have like variable substring lengths. Now, how do you overcome this limitation using a trie? So you want to use a trie, but you do not want fixed substring length. Now, let's see an example of how we can do it. And this will introduce the idea of indexing with suffixes rather than a fixed substring. So till now we used the substrings here and indices, and here also the substrings and the indices. These are fixed substrings, but these are substrings. Now what we'll use is we'll use suffixes rather than substrings, and we'll try to use indexes on them. Now, if you consider this following text, this big text, this is the text. What you do is you add a dollar at the end of the text. So this is your text. You add a dollar like this, and for so if this is t and you add your dollar, so this is t dollar. A list of substrings of this t dollar sequence will look something like this. So this whole thing is a substring of this whole thing. Just if you omit the g, then you omit the next t, then you omit another t, then like you remove one alphabet from the beginning, so it will become a substring of the previous string, right? So this is how you generate a list of substrings which are suffixes. So what are suffixes? The end part of the sequence, right? So if you remove elements from the beginning, the result what you get is basically a suffix of the previous string, right? So so you keep on building suffixes like this. So for a string like this, all suffixes will look like this, right? Now let's use the rules to build a suffix trie for a much simpler text, which is this small text like ABA. It's a very simple text. We will use this to build a suffix trie, right? It is much smaller than the text that we use, so it will be easier to get the point across. So as usual, as said, we add a dollar sign at the end, and we generate all the suffixes for this particular string. So a dollar, then a dollar, a dollar, like this. You generate all the substrings. Now let's try to construct the suffix trie. So this is the trie here. Again, you start from the root. Again, you start from the root, and you move along the edges of the nodes, and you try to spell out the whole substring, right? So this is how it will look like, right? So if this is a root, uh, this is one substring a b a. So this is this substring. Again, say for this is this a dollar is this substring, right? This is the a dollar substring, and this is only the dollar substring. So if you notice, the the biggest substring of all, which is the whole text itself in this case. So ABA dollar. So this is the longest suffix. So the longest suffix of a particular sequence is the sequence itself, right? So this is the longest suffix, and this is the shortest suffix, right? containing only the terminal character, doesn't have any other sequences, and your longest suffix will have the entire sequence. Now, now you can like make the try out of these substrings and suffixes. Yeah. Uh, yeah. So you have created this try using the suffixes. Now we want to see one thing. Like if you remove the dollar sign from it, if you remove this endpoint delimiter from the sequence, the try will look very dramatically different. Okay. The difference that without the dollar sign, the try will not have every suffix to be represented by a path from a root to a leaf. Right? So you will not have a possibility to represent every suffix from the path of the root to a leaf using a single, like, say for example, how many suffixes are there? 1, 2, 3, 4, 5, 6, 7. So there are 7 paths of this try from the root to the leaf. So this is 1, 1, 2, 3, 4, 5, 6, and 7. So using the dollar sign, it will give you, if you have n number of substrings, you'll have n number of paths connecting from the root to the leaves. But if you do not have that, if you do not have that end dollar thing, you will not have the exact same number of paths as the number of substrings or the number of suffixes. That becomes an issue because it'll create overlapping substrings, which will be hard to analyze and hard to use indexes on. So that's why you use the dollar sign at the end, which will give us all distinct paths from the root to the leaves, spelling out the suffix for that particular sequence. Right. Now, in suffix tree, each edge is labeled with a single character and the nodes have no labels in this particular type of representation. However, you can think of every node as being labeled with a string that spells out all the characters occurring along a path from the root to up to that node. So for example, this is that suffix try. Now let's consider this node here, and inside this node, you represent the sequence that comes from the root to that particular node. So for this node, it will be BAA. Say for example, this node, this will be AB. Again, for this node, it will be AAB. Right. So excuse me. So suffix try is an ideal data structure to quickly check if a pattern is present or not. Suppose we want to check if a substring, a particular substring, say BAA, is present within the text given, represented as a suffix try. We start at the root, we take the edge labeled with B. We start at the root, we take the edge labeled with B, uh, and we proceed to the to the proceed through an edge labeled A. At this point, there are two outgoing edges, A and dollar. Since the last character in BAA is A, we take the edge labeled as A. Right? Because the entire substring BAA can be spelled out as a path from the root, so we conclude that BAA is indeed a substring of ABAB. So what happens is, like, so B, so this is your BAA's sorry, ABABA substring, which is the entire sequence. You want to find if there are BAA substring present inside the uh, whole uh, text. Now you start at the root and you take this B A. So you take this. You start at the root. You take a A. I just want to. Okay, so this is your longest path, right? This is your longest path, which is telling you the entire sequence, entire text that is present. Now, in the longest path, uh, we start at the root and take a path that is labeled as B, and then you proceeded with a path labeled as A. Now there are two possibilities. There's you either go here or here, right? Now, since the last character in BA is A, you take the label edge labeled as A. Now the entire substring BA can be spelled out uh, within a path connecting the root to the leaf, which spells out the whole uh, text. So this is how you can know that this particular sequence is present in the is actually a substring of the particular text. So I think this is a better notation. So BA, then you move here because there's a dollar here. Now this is a whole substring that is present in your try. So you can be sure that that particular substring is present inside your text because it is present inside the try of your text. Right? Okay. Now we talked a lot about tries. Now what is actually a tree? A tree is nothing but if you just collapse all the non-branching paths and concatenate the labels. Say for example, you collapse these three paths here and you concatenate the labels like this. So this is ABA dollar. You collapsed all three, 1, 2, 3, 4 into 1 to ABA dollar, and then you collapse these two into 1 to BA like this. So if you collapse these non-branching paths and concatenate the labels and create the data structure, what you'll get is basically a tree. So trie is a much more comprehensive version of a tree, but a tree is a better representation in terms of space and everything. But yeah, it's very similar. Try it. So do not get confused, but like, the workings will be very similar. Okay. Now, if you are looking for a substring in tree, now you have seen how you can get it in a trie. Now let's see if you can get it in a tree or not. Now, the logic is exactly the same. As I was saying, trie and trees are very similar. So the logic to find, sorry, so the logic is exactly the same. Just the logic is exactly the same as with suffix tries, except now we have to account for the fact that along some edges, only a portion of the characters within a label will match, because we have like collapsed all these single edges into one. So it is possible that after BA, it doesn't match the whole thing; it matches only a certain portion of it. Right? It is possible. Earlier in a try, since there is only one edge and it is representing only one character, so it's either a match or a mismatch. There is no in between. But here in a tree, it is possible that it only matches partially, it doesn't match the whole thing, right? So the, if you want to check uh, a particular substring is present in the text or not, the logic is very similar to that of a suffix trie, except that you have to account for the fact that along some edges, only a portion of the characters within a label will match, the rest will not. In this case, the matching characters are highlighted. So here it is. So you want to find BAA in this substring, right? So here it was very easy. BAA is here. But if you see in a tree, it's not directly there, right? BA is there, one A is there, rest of the string is not matching there. Another BA is extra here, it does not match with your pattern. So this thing you need to keep in mind. So here B capital A matches, and this capital A matches. Rest of the substring does not match. Small b A and the dollar does not match with the substring here. So here, this is the thing that you need to keep in mind in comparison to a try. Right? The logic is same, but this is one of the caveat. that you need to keep in mind. But if it partially matches, it means that it is a substring. But the pattern should be a full match. Like, get what I'm saying? So if it is BAA, it should fully match. BAA should be present, right? So it should not be BAB here, right? It should be BAA, and BAA should match the string, right? Okay. So BA matches along the first edge, along the blue path, and only A matches in the second edge. The conclusion is that BA is the substring of the text. Right? Now, we talked about different suffix tries and suffix trees, but what is the major application of this suffix tree? Right? The most important application of suffix tree is to find the longest common substring. Right? To find the longest common substring of x and y, you make a new string, which is basically you add these two strings with a special symbol in the middle and a special symbol at the end. Right? So you have a string x and a string y, and you like concatenate these two strings with a special symbol in the middle, like differentiating these two strings, and there should be a special symbol at the end. So these two are technically both terminal symbols. One is at the terminal of for the first uh, of the first uh, sequence, and another is a terminal of the second sequence, right? Okay. Now you want to find what is the idea? The idea is to find the common longest substring of x and y. Okay. Now you make a suffix tree for this particular element. So you have x like this, y like this. Now you concatenate x and y like this, right? So between x and y there is a special symbol, and after y there is a special symbol. So you have concatenated it like this, and now you you build a suffix tree. Right? So you build a suffix tree like this. This is how your suffix tree looks like, and you have the indices at the bottom. Now consider these leaves in offset 0 and 4. So basically this is offset 0, this is offset 4. These are suffixes of x. Offsets in 6 and 11. This is 11 and 6 is here. These are suffixes of y. Because you see, this is xy, and this is getting divided here. So this is basically 11 1, and the sixth one are basically suffixes of y, and the 0 and the fourth one. So this is the 0th and this is the fourth one. These are the suffixes of x. Now if you traverse the tree and annotate each node according to whether the leaves below it include the suffix of y, x, or y, or both. So you do this, this makes your work much easier. So you have this created data structure, you have all the information of where the particular suffixes of these two patterns are. Now if you are given a particular sequence, you just find that sequence and find the longest common sequence. If you're given two sequences, you can just concatenate them, make the tree, and like traverse like this and check if the substrings are present or not. Right? And the deepest node annotated with both x and y has LCS as its label. So the deepest node, so you are asked to find not the common substring between x and y. You are asked to find the longest common substring of x and y. So the deepest node uh, in this tree with annotated with both x and y. So this is the deepest node with x and y. Uh, this will have the LCS as its label. And if you check for time complexity and space complexity, it will be the order of it will be in linear order. It will be a if your length of x is x and length of y is y, it will be order of x plus y. So it's linear time and linear space. This is very important because if you think in terms of genomes, firstly, if it is a linear time to make a suffix tree, so if your x is given as your reference genome and y is given as a read, then if you can make this thing in real in linear time and linear space, which is also very important. This is a very good way of checking if a particular read is present in another one or not. So, like, if you are asked to find the longest common substring of these two sequences, so ideally the longest common substring between the reference and the read will show you where this read will align to, right? So in alignment, this is where it comes in handy. And the most important thing is here is the space and the time. Because it does it in linear space and linear time, this is very useful for us, right? Because as you remember, we started off with heuristic method, very brute force method, which took exponential time. So we have come down a long way from that. Okay. Okay. Next comes the concept of suffix arrays. We have seen suffix trees. Now we want to see suffix arrays. Okay. So when suffix tree allows sequence comparisons to be performed in nearly linear time, what the issue there is, they have a large memory footprint. At some point, this becomes a serious limitation, making the use of suffix trees practically impossible. Right? For example, indexing human genome using suffix trees will require about 46 or 47 GB of memory. So it's a very big amount of space for indexing a single gene. Right? So suffix array offers a more compact solution to represent the text suffixes. It specifies a lexicographic order. So it's lexicographic order. Like A will come first, then B, C, like that. Ordering of suffixes derived from the text T. Just a second. Okay. Yeah. Okay. So suffix arrays will offer a much more compact solution to test text suffixes. Right? So this is your text. You add a dollar at the end, uh, like you do. Now, same as suffix trees, you create a suffix array which will tell you, which will store the position and the string as well. So it will store this dollar is present in sixth position, $A is present in five, and like that. You break it down into substrings and suffixes, specific suffixes, and you track down where the label is present and store them in a suffix array. Because one does not need additional pointers required for tree representation, the suffix array has a significant smaller memory footprint. So, for example, a suffix array for a human genome will occupy around 12 GB, which is like one fourth of the uh, space that a suffix tree will take to index a human genome. Right? Yet, this is this is a suffix array. So, one thing to remember here. So, if you have m, uh, sorry, if you have a m length string. So if your length of your string is m, will have m plus 1 elements in your suffix array. So your suffix array will have m plus 1 integers. It has to store as integers. Okay. Now this we have brought down the time significantly using suffix tries and everything. We have brought down the time. We had space issues as well, but since like anyway, our human genome is very big, so space is always an issue. So we have using suffix arrays, we have brought down the space even further. So we are currently in a position where we have like a decent balance between the space and the time. It is faster, much faster, but it also doesn't take so much of space. We have found that balance, but still we want to go one step further and reduce the space. Right? That is where the Burroughs-Wheeler transform on the FM index comes into play. So the Burroughs-Wheeler transform is a reversible permutation of a string. So for example, this is a string. This is a given string. This is a given stream. What you do is you create all possible permutations of the string. So this is given. You bring the last element to the first, that is one permutation. Then you bring the last element to this to the first again, is another. Then you do this again. Then you have all these combinations, and these are all sorry, all these permutations that are possible here. Now, if you take the last column, create all rotations, and take the last column. The last column is the Burrows-Wheeler transform of the given string, original string, which is this. Now, you'll ask, like, what is the point? Like, uh, why are we doing this to just to rotate and everything? So there are many applications of Burrows-Wheeler transform. One of them is compression. We'll see how compression comes into play, but the other one is into indexing, right? So what we'll do is we'll create these rotations and save the last column, which is the Burrows-Wheeler transform. It will give us an idea about the indices of the strings of the each of the elements of the string. Now, since our DNA is repetitive, so you'll have like 5 A's together, 15 G's together, 12 C's together, like this. So what you can do is you do not need to store this whole thing. You just need to store how many A's are there, how many B's are there, like obviously in order, how many A's, B's, and C, whatever they are there, and then store that as a Burrows-Wheeler transform, and from that you can do you can get the original text from there and also get the indices of the original text from there as well. So we'll see how it is. So this is your string. You do all the rotations. This is the first, second, and every rotation you have done, and then you sort this in a lexicographical order, with basically dollar being the smallest. You sort this first column using the lexicographical order. Dollar is the smallest, and then A and B and whatever, and then you save the last column as the Burrows-Wheeler transform, and this thing is called the Burrows-Wheeler matrix. You save this the last column. There are three important features that actually uh, is an advantageous thing for BWT. Is it can be compressed because you can have repetitions here. You can just store how many B's. So here you can just store it as a 2B, and at the end you can store it as a 2A or something like this. Like you can use it to compress. You can use it to reverse and reconstruct the original string. If you have the Burrows-Wheeler transform, you can reverse it to reconstruct the original string, and it also can be used as an index. Now let's take an example. Say this is your string. So this was your string. Here, sorry. Yeah. This was your string. ABA, ABA. There's a dollar in there, right? Yeah. So this is your original string, and you have added a dollar at the end. And now also you have marked like which is the first element, which is the like there are multiple A's, so you have marked which is the first A, which is the second, which is the third, like this. So for B as well, if there are other elements as well, you'll mark them. Okay. Now this ranking is done based on the order of the characters in the text, which is the T ranking. The order of the ranked characters is preserved between the first column and the last column, right? So if you see, if your like last column is looking like with A3 first, then A1, then A2, then A0, then your first column will also look very similar. A3 will come first, then A1, then A2, then A0. So this order is maintained between the first and the last column, right? So this is a very important property of Burrows-Wheeler transform. Not only for A, you can see for B as well, right? So B1 comes before B0. Here also B1 comes before B0, right? Now, just a second. I'll be back in just a second. So, as I was saying, the order in the first and the last column are conserved, be it for this element A or this element B. So if it is A3, A1, A2, and A0, it'll in the last column it will be in the same order like this, right? In between there will be other elements, but the order of the individual characters will be same for the first and the last column. Okay, now let's rank these characters in order of their appearances in the sorted list of rotations. Right? Now this is the ascending order. If you sort this, and this is the ascending order, right? So you have A0, A1, A2, A3, and then B0, B1. So you've sorted it in using the first column, and this is your sorted BW. Right? Now this is your last column, this is your first column, right? This is what happened. Now, like what you want to see is basically which comes after what. So you're trying to reconstruct the whole sequence. You want to see which comes after what. So if you're asking the question, what comes after B1 from the last column. So the last column, which will have the B1, it will match to a first column having B1. So you need to find which of the first columns has B1, right? Which BWM row begins with B1. So since the first row begins with a dollar, you skip that. Then the next four rows begins with A, you skip that. You skip this row starting with B naught, you skip that again. So the row 6 is the column that starts with B1. Now let's see how we can reverse this BWT, right? Sorry. Because of the LF mapping property, we can also easily reconstruct the original text from BWT, right? So this is a BWT uh, last column, and this is your BWT first column. So start in the first row. F must have, the first column must have the dollar. Okay, so the dollar is the first column. So how can you get the first column from the last column? You just sort the last column, you'll get the first column, right? If you sort this, say this is your last column, you only have the last column for this BWT. You sort this. The lowest is dollar, then A0, A1, A2, A3, B0, B1, B. Right? Now, now start in the first row. You start with a dollar. L contains a character which is just prior to dollar, which is A naught. Right? A naught. Now the dollar contains the first row will contain the dollar, and the dollar wala row will contain A naught in the last one, right? Now, once you've got an A naught, it's basically reconstructing in reverse. So you start with the dollar. So dollar is at the end. Then dollar is your last element. Then after that, you find your second last, third last, fourth last, like this. You go on. Like, okay. So dollar is your last element here. Then you move to A naught, right? Dollar in the same row, you move to A naught. Now you check where in the first column is A naught present. It's here, right? Then what is the last column of the A naught? It's B naught, right? So after A naught will come B naught. Now where is B naught present in the first column? Next, it is here, right? So with B naught there is an A2. So with B naught there will be A2. Now A2, where it will be present in the first column, it is present here. So A2 is along with A1. So A2 along with A1 here. Now A1, where it is present in the first column, A1 is here. Then there is B1. A1, there is B1. Now B1, where it is present in the first column, it is here. There is A3. So this is the reverse of the characters we visited, and that is what is giving you the whole text. So you are reconstructing the whole text in a reverse order, right? But the procedure is simple. You start with the lowest. So first you have the BWT of the text, which is basically the last column. If you sort the last column, you get the first column. You have the first and the last column like this. You start with the dollar. You move along the row into the last column. You check what the element is there. Find that element in the first column. Check that what the element there is in the last column. Like this, you go on repeating this. So Start in the first row, F must have for dollar. L contains the character just prior to dollar, which is A naught, prior to dollar, which is A naught. A naught is LF mapping says that this is the same occurrence of A as first A in F, like this one, right? Jump to the row beginning with A naught. L contains character just prior to A naught. So A naught is just prior to A naught will be B naught, right? Because if you remember, you have like while creating the BWT, you have created all the possible permutations. So if P naught is like this, the previous element of P naught is sorry. If A naught is like this, the previous element of A naught is P naught because A naught came from the back. So A naught here. So what is the element prior to A naught? It will be B naught, right? So it's simple in that form. Okay. So you get, so you repeat all these things, you get the whole string back. Okay. This is a way with how it is shown. So you start from a dollar, you check for A naught in the last column. Now you start with a dollar in the first, A naught is there in the last with the same row. Then you move to the first column and see where there is A naught. Then you move along the row, you find B naught. Then you check for B naught where it is in the first row. Then you move along the row like this. You can like do that to find the entire text, right? Okay, so yeah, so today we covered what is alignment, what are different sequence variations, how do we score for alignments, different type of algorithms starting with brute force, blast, and then we saw how we can index and use hash tables, different type of trees and substrings and everything, right? This sorted list, hash table, and a trie, how you can use this to store different substrings and their indices and how this can help in alignment. Then we saw a trie and like how to construct a trie, how to look for substrings in a trie. Then we looked at different like suffix trie applications, how you can find this longest path, shortest path, and these stuff. And then we move from trie to a tree and look for substrings in a tree and finding the longest common substring in a tree, right? And then we move to a little bit of an improvement in terms of space from suffix trie to suffix arrays. And then at the end, we looked at Burrows-Wheeler transform and this FM index, how you can compress, how you can compress a character, compress a whole text into a small one, and then you can regenerate the text using the indices. Because for first and the last column, you can always recreate that indices, recreate the whole text from the last column, right? Okay. I think from from my part, I think this is all I had to say. Just one example of Burrows-Wheeler transform we'll go through before we drop off. So, one second. Okay. So you are asked to find the original sequence for this Burrows-Wheeler transform. Right? So first question is, what is the original sequence? How do you find it? Let's look at the solution. So this is your BWT. So for LF mapping, this is your last column, which is your BWT. And if you sort the last column, alphabetically sorting the last column, you can get the first column, right? Alphabetically sorting the BWT string. So this is your first column, this is your last column, and then you do that kind of mapping. You start from dollar, then you move to C, then like that. So you okay. So you can use this the whole like procedure as I just described to reconstruct this text from the BWT, right? And if your first column is like this, you can numerically transform this using, like, there are 5 A's like this, 5 C's, 5 G's, and 4 T's. So you can numerically transform this as like this. So this is where your space and everything is reduced, because you can the whole sequence you can numerically transform into a much smaller sequence, and you can store it, and you can also regenerate your sequence from that as well, right? Okay. Okay, I think that's all for today. So nobody joined. I'm not sure why, but this was the time. So I had to go through the class. Please look in if you have any questions regarding this class. If you're looking after I posted this on YouTube, so if you had any issues or anything, please mail me in this address so that I can follow it up in the next class. Right. So I'll just put it here. The fluency, just mail it here. Okay. So that's all. I think I'll be dropping off now. So thank you. Thank you.